Now the topic before us is movement of substances in and out of the cell. Some substances like carbon dioxide, which is a cellular waste, requires to be excreted out by the cell. Suppose this is the cell and carbon dioxide is in high concentration. Now we can see that we know that carbon dioxide cannot be seen. For the purpose of example, we are taking this, assuming this. And suppose carbon dioxide is in high concentration inside the cell, and outside the cell the concentration of carbon dioxide is less. As soon as there is difference in concentration of carbon dioxide inside and outside a cell, carbon dioxide moves out of the cell through the cell membrane. This is the cell membrane by the process of diffusion, that is from the area of high concentration to the area of low concentration. Similarly, we again assume that this is the cell and the level of oxygen inside the cell decreases as compared to the concentration of oxygen outside the cell. The oxygen enters the cell through cell membrane by the process of diffusion. Next, water moves in and out of the cell by the process of osmosis. These substances move in and out of the cell from the region of high concentration to the region of low concentration. It means that if the concentration is high in a particular area, it moves from that to the area where there is low concentration of the substance. Now we will understand the difference between hypertonic, hypotonic and isotonic solution on the basis of concentration of solute in the solution. Solutions are of three types: hypertonic solution, hypotonic solution and isotonic solution. We will understand the meaning of solute and solvent in the coming slide. Using these terms, that is hypertonic, hypotonic, isotonic, always involves comparison between two different solutions. Let us assume otherwise. Suppose we say that he is taller. How much taller? He is taller than him. That is, we are making comparison of two persons. Next, if we say that he is more beautiful than her, we are making again comparison of two persons. In the same way, when we are talking about hypertonic solution or hypotonic solution, there must be the presence of some other solution with whom the comparison has to be made. Cells have a very limited range of conditions in which they can survive. It is not that if the conditions are too hot, too cold, too watery, too salty, too acidic or too basic, the cells cannot perform in all these conditions. They can function properly if there is the condition which is suitable to them. So we found out what happens when you place the cell in a solution which is more salty, that is hypertonic, much more watery, that is hypotonic, or has an equal salt concentration, that is isotonic. So we will try to find out the position of the plant cell when it is placed in hypertonic, hypotonic or isotonic solution. Now we will first understand the meaning of solvent and solute. Solutions are mixture of composed of solute and solvent. Suppose this is a jar, this is water. We have put some salt in it. So salt is a solute and water is solvent. A solvent is a substance that dissolves a solute resulting in a solution. A solvent is usually a liquid. Now what about solute? Solute is a substance which is dissolved in another substance. In the example you have seen that the salt has been dissolved in water. Water is a mixture, it is a solution, mixed solution which is composed of water and salt, in which salt is a solute and water is the solvent in which salt or solute has been mixed. Now we will define or understand the meaning of hypertonic, hypotonic and isotonic solution. We will try to understand the meaning with the help of pictures so that we can easily understand the definitions. First, hypertonic solution. In this picture, you can see that these dots are solutes. These are also solutes. So, we are talking about hypertonic solution. The solution which is inside this, this black color covering is hypertonic solution. It has more solute in it as compared to another solution. That is, if we assume that this is the cell and the cell has less solute, whereas inside the cell solute is more. So the solution which is outside the cell is hypertonic solution. So next, hypotonic solution. This is the hypotonic solution in which the cell has been placed. You can see that solute is in low concentration as compared to the another solution, that is inside the solution, inside the cell solutes are in high concentration, whereas outside the cell solutes are in low concentration. So a hypotonic solution is the solution in which solute concentration is low as compared with another solution. Another solution means solution inside the cell. So now talk about the isotonic solution. If we assume that this is the cell and you find that the number of solutes inside the cell and the number of solutes outside the cell are equal. So outside the cell, this is the isotonic solution in which it has the same amount of solutes as compared with another solution, that is solution inside the cell. The number of solutes which are present in the solution outside the cell as well as within the cell are same. So whenever we are talking about hypertonic, hypotonic and isotonic solution, we are talking about the solution which is outside the cell. Now the next topic before us is what happens if the plant cell is placed in hypertonic solution, hypotonic solution and isotonic solution. These are three different situations. We will discuss the same one by one. First question before us is what is hypertonic solution and what will happen if a plant cell is placed in hypertonic solution, and what is the meaning of hemolysis? As already discussed, a hypertonic solution is the solution in which solute concentration is high compared to with another solution, that is solution inside the cell. All of us know that water molecules free to pass through the cell membrane in both the directions. That is, water molecules can come in and go out of the cell membrane. If a living plant cell is placed in the hypertonic solutions, assume that this is the cell and it has been placed in the hypertonic solution, then what will happen? The liquid surrounding the cell has much higher concentration of solutes and the concentration of water is much lower compared to inside the cell. You can yourself see that solutes are less inside the cell, water is more, whereas outside the cell solutes are more, water concentration is less. Unfortunately, these solute particles which are present outside the cell membrane cannot pass through the membrane to reach equilibrium. So ultimately, the water molecules can pass through the cell membrane. So more water will leave the cell. You can see here that these are the pink color lines. More water is coming out of the cell. Only a small water is coming inside the cell. So what will be the net result? The cell will shrink because more water will go out of the cell and only small amount of water will come inside the cell, and the net result is more water goes out of the cell and the cell will shrink. We call this cell contraction plasmolysis. What this situation is called? This situation of cell contraction is known as plasmolysis. Now we take another example of hypertonic solutions. When compared to fresh water, sea water contains a higher concentration of salt particles. All of us know that sea water has much more salt particles. So the sea water is a hypertonic solution in which concentration of solute is on the higher side. Freshwater fish cannot survive in sea water because the cells would leak water into the surrounding salt water and they would quickly perish from dehydration. Suppose we assume that this is the cell of a fish and the fish is freshwater fish which has been placed in the sea water. Suppose this is the sea water and this is the cell of a freshwater fish, and what will happen? The water will come out of the cell of the fish into the sea water. The contraction of the cell of the fish will happen and ultimately all the cells of the fish would leak water into the surrounding salt water, and ultimately what would happen to the fish? They would quickly perish from dehydration and ultimately it would lead to the death of the fish, as there would be dehydration, as more and more water will come out of the cells of the fish. Now the next question before us is hypotonic solution, and what would happen if the plant cell is placed in hypotonic solution? You can see from the picture that this is the hypotonic solution, this is in which solute concentration is low as compared with another solution. This solution is solution inside the cell. Inside the cell, solutes are in high concentration, whereas outside the cell, solute concentrations are in low concentration. When a plant cell is placed in a hypotonic solution, what will happen? Suppose this is the plant cell and it has been placed in the hypotonic solution. You can see here that solutes are in less concentration outside the cell, and whereas the plant cell has more concentration of solutes. Water is more outside the cell, whereas water is in less concentration inside the cell. So when a plant cell is placed in the hypotonic solution, water will come inside the cell by the process of osmosis. More water will come inside the cell. You can see here that rosy color pinpoints are there, 4, and only one arrow is going outside. It means that more water is coming inside the cell and only a small amount of water is going outside. So the net result is that more water enters the cell and the cell swells up, and coming of more water inside the cell builds up pressure against the cell wall. This is the cell wall. More pressure builds up against the cell wall because lot of water have come inside the cell. The cell wall then exerts an equal pressure against the swollen cell. In this way, the cell wall prevents the cell from bursting. We can say that as more and more water is coming inside the cell may burst, but the cell wall prevents the cell from bursting because it's also cell wall also exerts its pressure inside against the swollen cell. Now both tap and purified water are hypotonic. When an single animal cell is placed in a hypotonic solution, too much water rushes inside. Cell will grow in size first. It may be a question in the exam. What would happen if a red blood cell or a single animal cell is put in a hypotonic solution? The answer is that too much water will go inside the cell and the cell will grow in size and burst. We have in the previous slide we had seen that the cell wall prevented the cell from bursting. That was plant cell because it was having an extra covering, that is cell wall. But in the case of animal cell, cell wall is not there. Only there is plasma membrane or cell membrane. As the animal cells do not have cell wall, so when the water comes inside the cell in too much quantity, the cell grows in size and bursts, whereas in the case of plant cell, the cells do not burst. Now what is the meaning of isotonic solution, and what would happen if the plant cell is placed in isotonic solution? An isotonic solution is the solution which has the same amount of solutes as compared with another solution. In this picture you can see that the number of solutes outside the cell and inside the cell are equal. So the solution which is outside the cell is isotonic as it has the same amount of solutes. So what would happen if the plant cell is placed in isotonic solution? So the concentration of solutes inside and outside the cell is equal. The amount of water inside and outside the cell is also equal. So the amount of water going in the cell and coming out of the cell is equal. The net movement of water is 0 because the solutes are equal. Equal quantity of solutes are present in and outside the cell. So the movement of water in net is 0 as the amount of water which is coming inside the cell is equal to the amount of water which is going out of the cell. You can see here that water is coming inside from two sides and water is also going outside. So the net result of movement of water is 0. In this case, what happens to the plant cell? The plant cell neither grows nor shrinks. It will stay the same size. Neither it will shrink nor it swell up. Here is another example of isotonic. Let us take 2 cups of tap water. One is big cup, another is small cup. In this big cup, we take 100 milliliter of water and we put in it 2 gram salt. After dissolving it, if we pour out some of this water into this, if we put out this solution into this, the concentration of solute in the solvent will remain the same. That is, the concentration of salt in the water will remain the same. So the solution which is present inside this small cup is also the same as compared with the solution which is present in the big cup. So the solution is isotonic, which has been placed in the smaller cup because nothing has changed. The ratio of salt is 2 grams salt per 100 milliliter. The ratio will remain the same. Now we take another example of hypertonic and hypotonic solution. Suppose you like to drink cold milk having 1 spoon of sugar. When you do not put enough sugar in the milk, then it is hypotonic solution because it has less concentration and it just tastes like cold milk without sugar. But when you put too much sugar in the milk, say 4 spoons sugar in your cold milk, then it is hypertonic because the milk is now extra sweet. An example of isotonic would be perfect cold sweet milk that is not too sweet and is not too light. It is equal. Now we understand the meaning of what is the process of diffusion. Diffusion is the process of movement of particles, that is solutes, from a region of high concentration to a region of low concentration. In this picture, you can see that a purple dye has been put out in the water. Here, the concentration of dye is more. It has slowly spread from the region of high concentration to the region of low concentration, and ultimately the dye color has spread, purple dye color has spread in the whole water. Diffusion is a passive process. It is not an active process. It is a passive process, which means that no energy is needed in this process. Breathing in oxygen and releasing carbon dioxide out of the body during respiration is an example of simple diffusion. Carbon dioxide and oxygen moves in and comes out of the cell by the process of diffusion. So the diffusion plays a very important role in the exchange of gases and water between the cell as well as the cell and its external environment. Now, what is osmosis and what is the significance of osmosis? The process of osmosis takes place in liquids. Osmosis is a type of diffusion. The spontaneous net movement of water or solvent molecules through a selectively permeable membrane from a region of high water concentration to a region of low water concentration till the equilibrium is reached is called osmosis. In this picture, you can see that this is the semi-permeable membrane. So in this part, you can see that solute particles are in less concentration and water is more. And this is a semi-permeable membrane. So slowly and slowly, the water passes through this semi-permeable membrane as the water is in high concentration in this part of the jar, and slowly it passes through the semi-permeable membrane till the equilibrium is reached. You can see here that the concentration of water in both sides is now equal. Equilibrium has reached finally. Osmosis is a passive transport. It does not require energy to be applied. Examples of osmosis in our daily life is plant cells soaking up water. Now, what is the significance of osmosis? Osmosis is very important for living organisms. Next, unicellular freshwater organisms and aquatic plants retain water through osmosis. Next, absorption of water by roots also occurs through osmosis. It means that the plants take up water through their roots by the process of osmosis. Next, movement of water in and out of the cell occurs through the process of osmosis. Now we will do recap of this course. The cells were discovered by Robert Hooke in 1665. Cell is the structural and functional unit of life. A unicellular organism, which is also known as single-celled organism, and it is an organism that consists of a single cell, whereas multicellular organisms are those organisms which have more than one cell, and the number of cells in multicellular organisms may range from a few to billions. The lifespan of a single-celled organism is short because the various life processes such as excretion, digestion are done by a single cell. So the lifespan of a single celled organism is short because of the load of work they have to perform, because load of work on the single cell in case of unicellular organism is very high. The lifespan of a multicellular organism is longer due to the reason that the different cells carry out specific functions. The cells of different living organisms may differ in shape and size. Next, shape and size of the cells are related to the specific function they perform. A cell consists of three parts: cell membrane, nucleus, as well as cytoplasm. Cell organelles are the membrane bound structures which are found in the cell, the various structures which are found in the cell. The plasma membrane is a biological membrane which surrounds the cytoplasm of a cell. Plasma membrane is also known as selectively permeable membrane because it allows only a selective material to go in and out of the cell. Next, on the basis of the concentration of solute in the cell, solutions are of three types: hypertonic solution, hypotonic solution, and isotonic solution. If you like our video, please like, share, and subscribe our channel. Thank you.